Hello and welcome to today's session. This session focuses on assignment discussion, experiments in biology. In this video, we will explore membrane permeability and osmosis. You will learn how to investigate membrane behavior under different conditions and understand the key principles of conducting biological experiments as a trainee biomedical scientist. The unit is titled Experiments in Biology, awarded by Ascentis. It involves investigating membrane permeability and osmosis. You must complete 2 experiments and compile them into one lab report. Adherence to scientific structure and safe laboratory practice is essential. The assignment is ungraded with a pass or refer outcome. 2 biological experiments are required, each documented in a combined report. Follow a scientific structure approved by your tutor. Meeting all assessment criteria is necessary to pass. Safe laboratory practice is mandatory throughout. The report should be supported by accurate data and analysis. Act as a trainee biomedical scientist investigating membrane behavior. Experiments are linked to cell biology and human physiology. Scientific accuracy and correct techniques are crucial. Results must be data-supported and analyzed thoroughly. The aim is to explore membrane behavior under different conditions. Develop hypotheses and measure data safely. Understand and draw conclusions from collected data. Discuss and evaluate the data thoroughly. Produce well-structured scientific reports. These outcomes ensure a comprehensive understanding of the experiments. Investigate how temperature affects membrane permeability. Use colorimetry to measure pigment leakage from beetroot membranes. This relates to the phospholipid bilayer structure. The experiment addresses specific assessment criteria. Propose an aim and hypothesis to guide your investigation. Devise a suitable methodology for the experiment. Carry out the investigation safely, adhering to guidelines. Record results using tables and graphs for clarity. Evaluate the hypothesis based on collected data. Review any errors or limitations for future improvements. Investigate the effect of water potential on osmosis using plant tissue. Measure mass changes at different concentrations. This experiment relates to cell membranes and osmosis. Address assessment criteria through structured investigation. Propose an aim and hypothesis for this study. Devise a suitable methodology for the osmosis experiment. Conduct the investigation safely, following all procedures. Present data clearly using tables and graphs. Evaluate the hypothesis using the results obtained. Identify errors, anomalies, and suggest improvements. Use clear tables with proper headings and units for data. Plot graphs like temperature versus absorbance to visualize trends. Identify patterns and highlight any anomalous results. Ensure your data supports the conclusions drawn. This clarity is crucial for scientific communication. Discuss the accuracy and reliability of your data. Identify sources of error within your experiments. Evaluate the suitability of methods used. Suggest realistic improvements for future investigations. Link your findings to established biological theories. Your lab report must include a title and aim, present a hypothesis and scientific background, detail the method and risk assessment undertaken, include results, analysis, and evaluation. Conclude with references following the Harvard style. Complete a risk assessment before starting experiments. Follow all laboratory safety rules diligently. Use equipment correctly to avoid accidents. Dispose of materials safely and responsibly. Report any incidents immediately to maintain safety standards. Ensure all work submitted is your own. Reference all sources using the Harvard style. Do not fabricate or alter any data. Plagiarism and malpractice are strictly prohibited. Uphold academic integrity in all aspects of your work. Consult A-level standard biology textbooks for background knowledge. BBC Bite Size offers insights into cell membranes and osmosis. OpenStax Biology provides additional resources. Follow tutor-provided laboratory guidance for practical advice. These resources will aid in your understanding and execution of the experiments. Prepare thoroughly before conducting the experiments. Ensure all materials and equipment are ready. Follow the methodology closely for accurate results. Record observations meticulously during the process. Use this data to support your final report and conclusions. Analyze your data using appropriate statistical methods. Look for trends and correlations in your results. Use graphs and charts to illustrate findings. Ensure your analysis is thorough and scientifically valid. This will strengthen the credibility of your report. Conclusion: understanding biological experiments is essential for interpreting scientific data accurately and drawing valid conclusions. These experiments provide insights into the mechanisms of life, allowing researchers to test hypotheses, identify cause and effect relationships, and develop evidence-based explanations. By critically analyzing experimental design, methods, and results, students and scientists can ensure reliability, reproducibility, and ethical integrity in biological research. Ultimately, mastering the principles of biological experimentation strengthens both scientific literacy and the ability to apply knowledge to real-world biological problems. Questions? Reach out via WhatsApp at 074843616888. Our team is always happy to help. Thank you for your attention, and see you at the next session.